Hello everybody. Today we'll talk about the nervous system, the most important system of your body. You already know that two systems of your body are called the control systems. One is the nervous system and another is the endocrine system. So these two systems control the other systems of your body. Among these two systems, nervous system is called fast acting control system because it responds very quickly by electrical signal transmission. Endocrine control is done by the hormones, endocrine hormones. So this one is much slower. Okay, and nerve signal transmission is electrical signal transmission, so it is very fast. Okay, anyway, so first we'll talk about the functions of the nervous system, classification of the nervous system or divisions of the nervous system, different types of cells in the nervous system. In the nervous system there are two types of cells. Supporting cells are called the neuroglial cells and the main cells are called the neurons. So neurons are the main cells and neuroglial cells are the supporting cells. There are different types of neuroglial cells. We'll talk about that and their functions. We'll talk about the parts of a typical neuron. Then we'll talk about few structures we see very often inside the nervous system: tract, nuclei, gray matter, white matter. And then we'll talk about the reflex and the parts of a reflex arc. So your nervous system performs thousands of functions, but we can group all those functions into three types: sensory inputs, integration, and motor outputs. Sensory inputs, uh, let me give you a couple of examples. When you see something, like you are thirsty or looking for a glass of water, so when you see a glass of water, you have the retina in your eye, and retinal photoreceptors will receive that signal, the picture. So the photoreceptors in the retina, right? So receptors receive that signal first, and then the photoreceptors from the eye, retina of the eye, will send signal to the brain, and that is called sensory input. And brain or central nervous system will do what? Will integrate that signal. Integration includes it will receive the signal, so receiving and interpreting. Interpretation, analyzing, right? So that is the function of the integration center, which is the central nervous system. It will receive the sensory input and then interpret it, analyze it, and then it will do what? Will send a motor command. That is the last function, motor command. through the motor nerves to the muscle. Why to the muscle? Because after you see a glass of water, your eyes send signal to the brain for interpreting, analyzing, and what the brain or central nervous system does in this case. First, your brain will quickly uh, decide if it is water or something else, right? It could be acid or something, dirty water. So brain will quickly interpret that if it is water, is it clean? Is it drinkable? And then it will also measure the distance of that glass, the shape of that glass, because the brain will send signal to the muscles of your hand to reach to that glass of water and grasp it and then bring it to the mouth. So you see that all those analyzing and measurements, all those interpretation are done in the integration center in the central nervous system. And then based on those interpretation, your central nervous system sends motor command through the motor nerves to the muscles. And the muscle in this case is the effector, called the effector, because it will perform the action. So effector or effectors perform the action, okay, and receptors receive the signal, okay, and sensory nerves or sensory input, sensory nerves take the signal to the integration center, which is the central nervous system, and then motor nerves takes the motor output, the command, to the effector to perform the action. So this is one example how the nervous system works, and the functions can be divided into three groups. Divisions of the nervous system. We divide the nervous system into central nervous system or CNS and peripheral nervous system or PNS. Central nervous system or CNS includes two organs, your brain and the spinal cord. So these two organs belong to the central nervous system, and they are heavily protected. You know that your brain is protected by the cranial bones, skull, and the spinal cord is protected by the vertebral column. Peripheral nervous system or PNS includes cranial nerves and spinal nerves. Cranial nerves in human nervous system, there are 12 pairs, and spinal nerves are 31 pairs. Cranial nerves are attached to the brain. So if this is your brain, this is the spinal cord, okay, going down, and cranial nerves are attached to the brain. So these are cranial nerves. Okay, these are cranial nerves. How many? 12 pairs. And spinal nerves arise from the sides of the spinal cord, 31 pairs. Since they are attached to the spinal cord, they are called the spinal nerves. Now, cranial nerves are attached to the brain, and they could be uh, two types. Some cranial nerves are motor, some are sensory, and some are mixed. Now, the motor nerves take the signal out from the brain. So those cranial nerves take the signal out, away from the brain. Those are the motor nerves. And those cranial nerves take the signal towards the brain. Those are the sensory cranial nerves. And in some nerves, you have both types of fibers or branches, sensory as well as motor. Okay, so those are mixed type nerves. Okay, spinal nerves, 31 pairs. All spinal nerves are mixed type nerves because they have both sensory and motor fibers together. Okay, so spinal nerves are mixed type nerves. Now, you know that sensory nerves take the signal towards the brain, and that's why sensory nerves are also called afferent nerves, taking the signal towards the brain, arriving in the brain. A. Motor nerves take the signal out from the brain, right? Outwards. So, motor nerves are called efferent, exit from the brain. Okay, efferent. So, motor or efferent, sensory or afferent. So, that is how we classify the nervous system. First, we divide into, you see here, central nervous system, brain, and the spinal cord, and peripheral nervous system includes sensory or afferent, motor or efferent. Okay. Now, motor could be somatic motor and autonomic motor. Remember, somatic is voluntary and autonomic motor is involuntary. What is the difference? Let me give you an example. If you see some delicious food, right, uh, or smell delicious food, secretion of saliva in your mouth or gastric secretion in your stomach occurs, right? So when you see the signal, see the food, signal from your eyes go to the brain, sensory input. Then brain processes if it is delicious and if you are hungry, all those things. Then brain sends signal to the salivary glands and gastric glands, and those glands cause secretion. So that secretion is involuntary, autonomic, because you don't do that. It happens by itself. So involuntary or autonomic. Okay, motor coming out from the brain to the gastric gland or salivary gland. That's why motor, but involuntary. Now, uh, if something, you want to grab, you are thirsty, you want to drink water, so your brain sends signal to the muscles of your hand, but this signal is voluntary. If you think that you want to move your hand, you can do that, right? If you decide that you will drink, you can move your hand. So from the brain, the signal is going to the muscle of your hand, but what kind of signal is that? Involuntary signal. Voluntary signal. That means uh, depends on your will. So those are examples that signal are coming from the brain, but one is voluntary, another is involuntary. Now, let me give you another example, like, knee jerk or knee reflex. This is the knee, and you have patellar ligament here. And when you hit here with a small hammer, your leg shoots forward, kicks forward. Why? When you hit here, you have stretch receptors, and stretch receptors get activated, and receptors send signal to the muscles of your leg. But first the signal goes from the receptors here to the spinal cord, and then from the spinal cord signal goes to the muscles effector of your leg, and that kicks the leg forward. And you don't do it voluntarily. When someone hits here, your leg kicks forward involuntarily. So that is an example of involuntary action of the muscle. But if you move your hand to drink water, get a glass of water, that is voluntary, right? Muscle action. So muscle action could be also voluntary or involuntary. This is involuntary or autonomic. Make sense? So you got some examples. Now, autonomic or involuntary is further divided into sympathetic and parasympathetic. Just know that sympathetic activation is responsible for fight or flight response. Fight or flight response that releases adrenaline, which increases the heart rate, heartbeat, increases the blood pressure, increases muscle tone, right? So that is the activation of sympathetic branch of the autonomic nervous system, the response of the body. Parasympathetic activation does opposite. Parasympathetic activation relaxes the body. That means heart rate decreases, blood pressure decreases, right? Muscle tone decreases. So they are just opposite to each other. Let me quickly go over. So, central nervous system includes the brain and the spinal cord, and peripheral nervous system. First, we divide into two types. You see here, sensory nerves take the signal towards the central nervous system, towards the brain, and the motor nerves take the signal towards away from the central nervous system, away from the brain and the spinal cord. Okay, and motor could be somatic motor and autonomic motor, and autonomic could be sympathetic and parasympathetic. Okay, now two types of cells are found in the neural tissue. Neurons are the main types, and neuroglial cells or glial cells are the supporting cells. Neuroglial cells, although they are supporting cells, not the main type, but there are different types of neuroglial cells and they perform a number of important functions, including migration towards the injured neurons. Neuroglial cells can move towards the injured area of the nervous system. Why they need to move? Like, this is the brain. Okay, you got injury here, so cells die in this area, right? So some neuroglial cells, they will go there to clean up that area by phagocytosis, to remove the dead cells or microorganisms. Okay, how they will do that? Phagocytosis. So they engulf. So phagocytosis is an important function of some neuroglial cells. Uh, Guide migration of young neurons. When your brain is formed in the very early, in embryonic, just think about the early part of your life, a small amount of neural tissue uh, forms the brain, and then they multiply, the cells multiply and form different structures of the brain, right? So some neurons will migrate this way to form one structure of the brain, another group of neurons will migrate to this way, form another structure. So the neurons are formed by the migration of the young neuroglial cells. They will move to different directions to form different structures. Control the chemical environment by releasing some chemicals. It can regulate the chemical environment of the nervous system. Insulation, a very important function, uh, by covering the nerves or neurons. uh, Neuroglial cells provide insulation. Some neuroglial cells form insulation around the nerves or neurons in the central nervous system and peripheral nervous system. Uh, in next few slides, I'll just mention the name of important neuroglial cells and one or two important functions. Important neuroglial cells of your nervous system include astrocytes, most common type, and shape, star shape. That's why called astro. Site means cell, and they form blood-brain barrier. Blood-brain barrier. So that is the main function. Microglial cells are helping by engulfing dead cells or microorganisms. So phagocytosis is the function of the microglial cells. So they can engulf the microorganisms or dead cells. Ependymal cells are present inside the ventricles. Inside the brain, you have fluid-filled cavities. Those are called ventricles. And in the wall of the ventricles, you have the ependymal cells. We'll talk about that. So the ventricular walls are lined by ependymal cells. Oligodendrocytes, satellite cells, and Schwann cells. These three types of neuroglial cells form myelin. Or you can say these three types of neuroglial cells, all of them are involved in insulation. Covering the neurons. So the electrical signal transmission will be faster. At your home, you see the electrical wires are covered uh, by insulator, and that prevents the loss of electricity and makes the electrical signal transmission efficient. Okay, electrical transmission. So now you can think that if the insulation is lost, then the signal transmission will be what? Slower. Transmission. Okay. So those are the important neuroglial cells and their important functions. You see here, astrocyte looks like a star, and how they form blood-brain barrier. You see, this is a capillary inside the brain, and around the capillaries of your brain, you see an extra layer is formed by the processes arise from the astrocytes. So these processes arise from the cell body of the astrocyte, and they cover the capillary from outside. So why that is important? Because this is your brain and this is the capillary inside the brain. Okay, neurons, neural tissue, highly sensitive, right? Neural tissue is very sensitive. So you have blood here, and you know that blood has uh, many different types of chemicals, right? Some are good, some are toxic. Now, usually the capillary wall is very permeable. There are many tiny holes, so the chemicals can easily get out. But in the brain, around the capillary, the astrocytes form processes and covers the outer layer of the capillary like this. So an extra layer, this is astrocyte process, foot process that covers the outer surface of the capillary. So the toxic chemicals cannot easily get out from the blood. Okay, but in other areas of the body around the capillary, you don't have that extra layer, and that's why this layer is called the blood-brain barrier. between the blood and the brain tissue, this barrier. So that is formed by the astrocyte to protect your brain against the toxic chemicals from the blood. Uh, Microglial cells do phagocytosis. You see, they have many processes and branchings, and they can engulf the microorganisms or dead cells. Ependymal cells, inside the brain, you have fluid filled cavities. Like this is your brain again, and this is a cavity inside the brain called the ventricle, and the ventricles are filled with what? Cerebrospinal fluid, or CSF. So fluid filled cavity is the ventricle, and if you see the wall, the lining of the wall, you'll see the ependymal cells, and these cells have cilia, soft hair-like structures like this. So cilia arise from the ependymal cells. So these are ependymal cells, and they have hair-like things. Those are called cilia, soft hair, and they always move the fluid, so that can push the fluid away, and that flow of fluid is very important. So you see here ependymal cells, and these are cilia. Now you remember last three types of neuroglial cells I mentioned form insulation. So this is one of those three oligodendrocytes, and oligodendrocyte is the neuroglial cell. This is the cell body, this is the nucleus of oligodendrocyte, and you see from the cell body uh, uh, the extensions come out, and those wrap the nerve fibers. You see, make several layers around the nerve fibers. Here you see a number of layers covering around the nerve fibers. So oligodendrocytes, remember, have processes that form the covering, and that covering is called myelin sheath. It's white, lipid-rich structure around the nerve fibers, where in the central nervous system, remember that. So oligodendrocytes form insulation around the central nervous system neurons or nerves. Then other two types of neuroglial cells form insulation to satellite cells and Schwann cells. And this is a neuron of the peripheral nervous system, or PNS, not CNS. Okay, the last one was CNS, oligodendrocyte. Satellite cells and Schwann cells form insulation around the neurons or nerve of the peripheral nervous system. And these cells, you see, are the satellite cells, and they cover the cell body of the neuron. And these cells are Schwann cells. They cover the axon. So you know this is the axon, and they form covering or insulation around the axon. Now, I mentioned that the main function of this covering or myelin sheath is efficient. It makes the electrical signal transmission efficient. Now, what can happen if in your brain, central nervous system, this myelin sheath is destroyed? There is an autoimmune disease that is called multiple sclerosis. It is an autoimmune disease. Your own body produces immunity against your own body structures. So, uh, you know that you have macrophages, right, in the tissue. They do phagocytosis of microorganisms and uh, dead cells. But if autoimmune disease occurs in the body, in multiple sclerosis, what happens? These macrophages get more active, hyperactive, and they start to destroy the myelin sheath, okay, engulf the it's it up the myelin sheath. So that is called demyelination. Now, what will happen if demyelination occurs? The myelin sheath is destroyed, then the signal transmission, electrical signal transmission, will get slower. So the brain will get slower. The whole nervous system function could get slower. So first sign starts like blurring of the vision, or eventually loss of vision can also occur. uh, uh, Fatigueness, muscle paralysis, all those symptoms are uh, the outcome of this multiple sclerosis. So now we'll talk about the main cells of the neural tissue, are the neurons or nerve cells. Now, although neurons are highly sensitive, uh, and uh, even if you open the skull, the brain tissue may start to degenerate or deteriorate in touch of air. So it is highly sensitive. Uh, However, if you can protect them, the neurons are very long lived, can live more than 100 years. Okay, so the lifespan could be very long if you can take care of them. Okay, that's why neurons or neural tissue is protected by the bones. Amitotic. A means absence of mitosis, cell division. So neurons, if die, neurons cannot, no new neurons are usually produced because multiplication or mitosis usually does not occur. There are exceptions, but usually mitotic. Other cells in the body, that can quickly multiply, like your skin or muscle. Skin cells are dying all the time. Inside the mouth, the epithelial cells are dying, right? When you drink something very hot, cells die in the tongue, in the cheek, but they quickly regenerate. Red blood cells are dying after 3 or 4 months, but new blood cells are being produced. But once the uh, neurons in your uh, brain or the spinal cord die, usually they are not replaced. So that is a big problem, right? If someone gets lesion in the brain, you see that the symptoms don't get back. The normal function usually doesn't comes back. Uh, Neurons are long-lived, but they need continuous supply of oxygen and glucose. That means oxygen and nutrients. If I stop the blood circulation to your brain, the brain will quickly die, right? You know that if I press the neck to stop the circulation to the brain death occurs very quickly. Why? Because that will stop the oxygen and glucose supply to the brain. But if you stop the blood circulation to other organs of the body or muscles, um, it will not quickly die. Electrical signal transmission is the way signal is transmitted through the neuron. So neurons transmit by electrical signal. Parts of a neuron, if you see the structure of a typical neuron, this is a typical neuron, and a neuron has a cell body. So this is the cell body here. Inside the cell body you have a nucleus, and around the nucleus in the cytosol or in the cytoplasm you have nissl bodies, those dark structures, also called nissl granules, same thing. Okay, nissl bodies or nissl granules. Um, they are highly concentrated endoplasmic reticulum, and then from the cell body you see a number of short branching processes arise, and these are called dendrites. So dendrites are short branching multiple processes, and also from the cell body a single long process arise, and that is called the axon. And the area from where the axon starts, that area is called axon hillock. So this area or part is called the axon hillock, where the axon is attached to the cell body. Okay, so processes are dendrites and axon. Now the end of the axon, if you see the end part, there are a number of branches, and these are called axon terminals. Makes sense? Ends of the branching ends of the axon terminals. And the axon is covered by, in the peripheral nervous system, you have seen that the axon is covered by um, the Schwann cells, one type of neuroglial cells. We talked about that. Schwann cells form insulation or myelin sheath around the axon of the neuron. However, in between the Schwann cells, you see this is one and this is another. In between, the short spaces are uncovered, and these uncovered spots are called node of Ranvier, nodes of Ranvier. This is one node, so these are the nodes of Ranvier, where the axon is not covered or not myelinated. Okay. Um, Now this is a typical neuron that has cell body, nucleus, nissl granules, and multiple short branching processes are called what? Dendrites. So these are dendrites. Okay. So dendrites, single axon, axon terminal, These are axon terminals and Schwann cells, right? Form myelin sheath, insulation, and in between Schwann cells you have node of Ranvier. So this is SCHWANN, Schwann cell, and in between Schwann cells you have open spots called node of Ranvier. Okay. Now, one thing is very important here, that dendrites bring the electrical signal into the cell body. So electrical signal travels this way through the dendrite, enters into the cell body, then cell body will process the signal, and through the axon the electrical signal will get out from the cell body. So electrical signal will get out through the axon. And now another neuron is next to it. So this is another neuron, and you see here what happens. The dendrite of this neuron receive the signal from the axon terminal of the previous neuron. So this is the dendrite of this neuron, and this is the axon terminal of previous neuron, and this is a synapse where they talk to each other. The axon terminal of previous talks to the dendrite of next neuron. So this is called neuron to neuron synapse. Neuron to neuron synapse. Okay, makes sense. And then dendrites will bring signal into the cell body, and then cell body will send signal out through the axon, and you have then another neuron here. Again, the dendrites take the signal from the axon terminal of previous neuron, and that is synapse. Okay. So now also synapse could be between the neuron axon terminal and the muscle fiber. You already have learned about this. This is a muscle fiber, and this is the axon terminal, and this is called a neuromuscular junction. So axon to muscle, axon to muscle fiber, and this is called a neuromuscular junction. And neurotransmitters are released from the axon terminal here. You see a gap, that is the synapse, and neurotransmitters are released here at the synapse. So from the axon terminal, neurotransmitters or neurochemicals, neurotransmitters. Now neurotransmitters could be excitatory or inhibitory, or inhibitory. Now in some synapses, most of the synapses, excitatory neurotransmitters are released, so the signal will get stronger excitation. And if inhibitory neurotransmitter is released here, for example GABA, which is an inhibitory neurotransmitter, that will slow the transmission here at the synapse. Okay. You remember I said in the nervous system often we see white matter, gray matter, tract, nuclei. What is white matter? If this is, again, this is the brain, and the outer surface or outer thin layer of the brain is called the cortex. Cerebral cortex is the outer layer. Okay, and cerebral cortex is a gray matter. Okay, why? If you see under the microscope, the cell bodies of the neurons, you remember the structure of a neuron, the cell bodies of all many neurons are there, and also from the cell body the dendrites, right? You know that the dendrites are short branching processes are here, and then axons. Okay, these are the axons of those neurons. Now you must remember that I told you that in the cytoplasm of the cell body you have dark granules called the Nissl granules. So all these cell bodies are filled with that Nissl granules. So now you can think that when you see this area, it looks gray. Now let's see this area. All these axons are covered by white myelin sheath insulation, right? So when you see this area, since all white, covering of the axons, this area looks what? White. So this is called the white matter. So in the brain, outer layer, which is the cerebral cortex, is a gray matter, looks gray, and inner part looks white matter. So if you cut the brain, you can clearly see the gray matter and white matter areas. Now, uh, in some areas of the central nervous system, you'll see that many cell bodies or neurons or cell bodies of the neurons are highly concentrated like this. Not everywhere. In some areas, they are very densely packed, and these are called nuclei. So if you cut the brain, you can see these areas are even more gray. So nuclei are the areas where you have more cell bodies than the gray matter. Gray matter has all the cell bodies, but in nuclei you have more densely packed cell bodies. Okay, and in some areas in the brain you will see that the axons are heavily packed. Here you have all the axons, right? But some cases you will see that axons are more densely packed, like this one has densely packed axons, and these are called tracts. Okay, tracts, bundle of axons. Okay, so in gray matter you have the cell bodies and dendrites. In the nuclei, you have cell bodies and dendrites, but more densely packed. That is the difference. In white matter, you have myelinated axons, but in the tracts, you have myelinated axons, but more densely packed. That is the difference. Okay. Now, that is in the central nervous system. If you see in a nerve, like a peripheral nerve, a round structure like this is present, and that structure is filled with the cell bodies of neurons in the peripheral nervous system, not in the central nervous system. Okay, this is called a ganglion. Okay, so nuclei and ganglia is plural, same thing, ganglion, but nuclei are in the central nervous system and ganglion is in the peripheral nervous system. And similarly, uh, these are the nerve fibers, axons, right? Myelinated axons in the peripheral nervous system. And when the bundle that forms a nerve. So in peripheral nervous system, it is called bundle of axons, is called a nerve. In the central nervous system, bundle of axons is called a tract. Okay, so those are some structures we often see in the nervous system, central nervous system and peripheral nervous system. Now, uh, let's talk about different types of neurons. The neurons I showed you before, the picture or I did draw, that is a typical neuron. Most of the neurons in the nervous system are like that, and those are called multipolar neurons. It has a single axon and several dendrites, right? Most abundant. Bipolar neurons, if you see the neuron, this is the cell body, this is the axon, long, and only one dendrite. So if you see one axon and one dendrite, that is a bipolar neuron. Okay, unipolar, also pseudo-unipolar, uh, doesn't have any dendrite, only an axon. So this is the cell body nucleus, and actually the cell body is attached to the side of the axon. This is a single axon, no dendrite here, going like this. Okay, so single axon and cell body is attached somewhere in the axon to the axon, and that is called uni. Uni means one, unipolar or pseudo-unipolar. So multipolar is like this. I did already draw before. Multiple short branching dendrites, that is multipolar. Most common type. This is bipolar, one axon, one dendrite, and only one axon is unipolar. So those are different types of neurons. You see these are multipolar, one axon, many dendrites, bipolar, one dendrite, one axon, one dendrite, one axon. This is only one axon. Okay, so this is the cell body. So this is unipolar. I already talked about the synapse, where the axon terminal talks to the dendrites of next neuron, that is neuron to neuron synapse, and neuron axon terminal can also end on the muscle fiber, that is neuron to muscle, and that is the neuromuscular junction. That is another type of synapse. Neurotransmitters are released into the synapse, and some are excitatory to enhance the transmission, some are inhibitory to slow down the transmission. And by the action of those two, the nerve transmission is controlled or regulated. And up or down regulation of neurotransmitters can lead to many clinical conditions. Lack of neurotransmitter or too much, neurotransmitter can lead to many different types of neurological or psychological disorders. The last thing, reflex. Reflex is an autonomic response to a stimulus, and I have already given you an example. Your patellar ligament is here. If I hit here, tap here with a tiny hammer, the leg kicks forward, and this is involuntary. If you do voluntarily, that is not a reflex. Okay, involuntary one is the reflex. So must be involuntary or autonomic. Number two, every time when it responds, it will move this way. So the response should be same way. Okay, uh, same response. It is not like, sometime it will go this way, sometime it will go this way. No, always in same direction, to the same direction. So involuntary, autonomic, same response, and rapid. The response is very fast. So those are important properties of a reflex. Okay, to be a reflex, it must be autonomic. It must respond same way every time and must be very rapid. Another good example of reflex is pupillary reflex. You know that pupil of the eye. If I put flashlight, the pupil constrict, right? And every time I'll put flashlight, the pupil will constrict, cons constrict. Constriction of the pupil will get smaller, right? To limit the entrance of the light. So you don't do that. It happens by itself when light falls on the eye, and every time it will get short, not sometime big, sometime short. So same type response and immediate, very fast. Now, to complete a reflex, you need these components. First, receptors. I told you that, when you hit here, you have the patellar ligament has the receptors stretch. Receptors. So receptors must be activated first, right? Otherwise, the signal will not travel to the central nervous system. Okay, so sensory neurons, you already know, that takes the signal towards the central nervous system, where integration occurs, interpretation occurs. Then motor output comes out. We talked about this at the very beginning of today's lecture. So motor output, and the muscle moves the leg, that is the effector, performs the action, right? So receptors, number one. Number two, sensory input or sensory neurons, and then integration in the central nervous system, motor output, and last component is the effector who will perform the action. Okay, so those are the parts of a reflex to complete the arc. The arc is the whole circuit from the receptor to the effector. This whole circuit or pathway is called the reflex arc. Okay, so hope you uh, go over these things again and again, and you spend a lot of time to go over. Thank you.